So I'd like to begin also by thanking the organizers for the opportunity to speak. I really appreciated and enjoyed David's talk a lot, and Brendan's, and now Gautam's. I think that our work succeeds in an ecological succession, what has been discussed. So I'm going to talk about how antibiotics perturb the early life microbiota, fueling obesity. And I picked this topic among a number of other ones because the story is becoming the most clear. So, as David implied, our acquisition of the microbiota is vertical. We get a lot of our early life microbiota from our mothers, and they from their mothers. But mothers today are not the way they used to be. They live in a world of antisepsis. They've received antibiotics before and during pregnancy. There are many dietary additives. And babies aren't the same as they used to be, because in the United States, a third of them today are being born by cesarean section. And there is extensive bathing and formula feeding, and of course, early life antibiotics. And all of these potentially or actually impact the conserved intergenerational transfer of the microbiota. And so about 5 years ago, Stan Falco and I hypothesized that there is an effect of maternal status on the resident microbiota of the next generation, in which, as shown here, it is stepping down across generations. It's the kind of extinction that Brendan has been talking about, in which the effects on one generation are cumulative across generations. And in fact, there is a growing body of evidence that this is true. Now, I want to focus on obesity. Most of you have seen maps of this, like these CDC data, showing that in 1989, there's no state with more than 14% of adults obese. By 2010, there's no state with less than 20% obesity. What's happened is happening everywhere across the country. You see the map. It's also happening across the world. And it's only 21 years. Something very powerful must be going on. What is of sufficient scale? Well, one thing that is of sufficient scale is antibiotic use. From recent CDC data, they surveyed antibiotic use in the United States. In 2010, there were 258 million courses of antibiotics prescribed, 833 per 1,000 population. That's 5 courses for every 6 people, and the CDC data indicate that this has been going on year after year. The rates are age-specific. The highest rates are in young children, as shown on the left-hand side of the slide, and it goes down with age. And if you do a back-of-the-envelope calculation, you can calculate that the average child in the United States gets about 3 courses of antibiotics in the first 2 years of life, about 10 by the time they're 10, and about 17 by the time they're 20. These are consistent with many other studies. So there's extreme antibiotic exposure just from prescribed antibiotics, not to mention other factors. Now, here's the CDC map of antibiotic use by region. National average 833, Northeast and Midwest similar to the national average. West and South quite different. West lower, South 50% higher. No 50% difference in serious bacterial infections. Now it's interesting to look at the 2 CDC maps together, the geography of obesity and the geography of antibiotic use. Very strong pictorial representation. This is observational data. We don't know what the causal relationships are, but it certainly gets our attention. Now, as Gautam and Brendan pointed out, farmers have known for a long time that antibiotics change development. That's why they are so widely used. They use sub-therapeutic antibiotics. They've shown it works across species, not just mammals, across antibacterials, and the earlier you start it, the more profound the effect. So we began studies in mice. I'm going to just show you a couple of examples of our experiments in mice to test the question of whether antibiotics change development. The first study I'm going to show you was started by Il-Sung Cho when he was a postdoctoral fellow in my lab. He's now on the NYU faculty. Here, Il-Sung compared 4 different antibiotic regimens versus control. The antibiotic regimens changed body composition. There was an increased body fat. In another study, Laurie Cox looked at the systemic effects and hepatic effects of STAT shown by these MRIs. On the top panel, we see that there's increased fat, there's increased liver fat, and we see that there's increased expression of many critical genes in energy processing. In another study, Lori, which we called FatStat, the effect of combining high-fat diet and antibiotics, Lori started mice on antibiotics or not. They were on normal chow for the first 17 weeks of life, and then subsequently, half of them were put on a high-fat diet. Here are the results in male and female mice. From the left, total mass, fat mass, lean mass. If you look at the males, total mass, the bottom line is control group, no antibiotics, normal chow. Next line up is antibiotics, increase in mass, just like on the farm. Next line up, high-fat diet, the effect of high-fat. Top line, high-fat plus antibiotics. If you go all the way over to lean mass, you see the antibiotic effects. And if you go in the middle and look at fat mass, most of the fat comes from being on a high-fat diet, but it's even more on antibiotics. And now if you shift down to female mice and look at fat mass, the average female mouse on a high-fat diet put on 5 grams of body fat. With the antibiotics, they put on 10 grams. It doubled the amount of fat. It potentiated the effect. So one of the questions we had is this, do you need lifetime exposure, or is there short exposure sufficient, even 8 weeks or 4 weeks? So here we're looking at the effects on phenotype. The black line is the control. All 3 antibiotic groups had the complete effect. 4 weeks was sufficient. So what about the microbiome? At 3 weeks, there are only 2 groups, getting antibiotics or not. In the principal coordinates analysis, no antibiotics is in black, antibiotics is in orange. They're separated. There's an antibiotic effect. At 8 weeks, there are 3 groups, antibiotics or not, or stopped antibiotics. Black are the controls, orange are the antibiotic, and blue are the mice, the microbiota in the mice that received antibiotics and stopped. They have reverted to normal. So the perturbation was transient. But in the upper right-hand corner, you can see that the effect on the phenotype was permanent. This is evidence for a developmental effect. We see big changes in gene expression in the ileum across experiments. This is very much a function of both innate and adaptive immunity as well. To finish, we can think about the child who's born as an individual who's still developing. They still have stem cells and stem-like populations. They have to determine what's their fate. If they're mesenchymal cells, are they going to become fat, bone, or muscle? We focused on diet and calorie. We think that microbes in the cells that they're signaling through provide the context for these developmental signals. That's how it used to be. This is how it is now. Antibiotics and other factors are changing the founding bacterial populations. They're talking differently to the cells that they usually talk to, and these are creating a different context for development. Thank you.